আমাকে এই সুযোগটা দেওয়ার জন্য আমি ডক্টর দণ্ড এবং বাংলাদেশের সবাইকে অজস্র ধন্যবাদ জানাচ্ছি, কারণ এই প্রথম আমার ইনফ্যাক্ট এটা আমার প্রথম অনলাইন মিটিং বলতে পারেন। তো আজকে আমি ওই ইনেট ইমিউনিটি নিয়ে বলবো। তো এর মধ্যে কতগুলো পয়েন্টস যেগুলো আমি ডিসকাস করব, তার মধ্যে রয়েছে ইভলিউশন অফ হিউম্যান ইমিউন সিস্টেম, features of innate immunity, mode of functions of innate immunity, mucosal immune system, inflammation, complement, cutaneous immune response, granulocytes in immune response, toll-like receptors in immunity, এবং কি করে ইমিউনিটি গুলো কাজ করে সেগুলো আমি ডিসকাস করব। So the evolution of human system has been through billions of years, and what you will see here that the first one started with bacteria 2.4 billion years ago, and that is the time when it first tried to live on the eukaryotes. And during this interaction between the prokaryotes and the eukaryotes, these different viroids and viruses had exchanged the genes through both bacteria and eukaryotes. So this gene transfer resulted in coevolution of both the organisms. And what we see is, importantly, that I wanted to show here is, till the Silurian period, you see, when we first got the gnathostomes, means the animals with jaws, everything, the whole immune system was only complement and other innate immunity proteins, but there was no MHC, no B cell receptor, no T cell receptor, no recombination activation genes, or terminal deoxyribonucleotide transferase, which are required for generation of the diversity and wider specificity for the antigens, for these antigen-specific receptors DCR and TCR. And all those things you will see in Silurian period, which is called the Precambrian, post-Cambrian explosion. All the genes appeared at the same time. So what actually caused all this explosion at the same time is not known. But basically what we see here, the first point is that innate immune system components came first, and then at a later period, almost after 1,000 million years, we got the system for antigen specific recognition through like MHC, B cell receptor, T cell receptor. কালকে ডক্টর দণ্ড এবং ডক্টর চৌধুরী, ডক্টর সুজাতা গাঙ্গুলি, এরা সবাই এই ব্যাপারে খুব সুন্দর করে বলেছেন। So basically after that, through different phyla and classes of animals, we arrived here at the top, human, which is about 60,000 years ago. And this is the most complex immune system that the animal kingdom has. So what happened during this period of coevolution? Let us say that at the beginning, we had the two organisms, both are unicellular, but one was stochastically weaker than the other one, which was stronger. And obviously that weaker one tried to live or encroach or make the other stronger one as the host. So let's say this was the viroid-like RNA particles or the parasites, and they tried to live on the stronger one, which was self-sufficient. And then you see the complexity, which was very simple here. It grew complex in the later portion and during the course of evolution, RNA elements coding proteins, and then further. And during this period, different retroposon elements evolved. And here also we had the increase in DNA genome size and the complexity of the RNA and DNA elements, their arrangement, their regulations, everything was more and more complicated. And that is how finally we arrived at what we have here. And what we see here is that one side is the virus world and the other side is the metazoan multicellular animals world. And I think it is still evolving, and that is why we keep getting different viruses or different pathogens, and viruses in particular because they can evolve very fast, which is not possible for the animals which have the sexual reproduction. And therefore, I think COVID is not the last one. We will continue to see many different viruses in future. So basically, as it is said in biology, ontogeny recapitulates phylogeny. So this is what the development of human immune system. So during the first 9 to 10 weeks, you see here in both the myeloid and the lymphoid compartments, as introduced yesterday very nicely, the macrophages, tissue resident, the monomac cells, the monocytes, mast cells, dendritic cells, and then in the lymphoid, different innate lymphoid committed progenitors, and then NK progenitors, B cell progenitors, they are all there. So the innate immune system cells are coming first, and then these cells are preparing the progenitors, and then 10th week onwards, you see all these different cells have started populating the immune system. What is very important in this case, this 39 to 48 week during this birth, basically the humans have the T-dependent system and the T-independent system both fully mature, which is not the case in the model systems like in mouse, which takes about another 3 weeks to mature after birth. So the humans are mature at birth, human immune system. And what we see here is that during the earlier period, I have made here a bit position here. You'll see this is the time when this is the tolerance establishing zone, tolerance to maternal antigens, microbial metabolites in utero infections. And later on, we keep accruing in different establishing functional immune responses against microbiota or any perinatal infections. So what we get is basically during this development, both innate and adaptive immune system. What I have tried here is that the innate immune system has these different pathogens like virus, bacteria, parasites, and fungi, and they express different molecules on their surface, which we call pathogen-associated molecular patterns that act as antigens, and they are recognized by innate immune receptors, and they lead to the production of non-specific effectors, which reduce the pathogen load. And also these antigen-presenting cells, which have the intracellular pathogens or can engulf the extracellular pathogens or their antigens released by them, can present that antigen to the T cell compartment. So the T cells are presented with that antigen, and some of the antigens are also recognized by the BCR or B cell receptor, immunoglobulin alpha and beta chains. And these, with the help of T helper cells, these B cells are activated, and then we have both the effector B cells, which produce antibody, or the plasma cells, which secrete antibody, and the T cells, which secrete different cytokines, which Dr. Chaudhary is going to speak after me. And in addition, we have the memory B cells, and these memory B cells participate during the second exposure here. And during this second exposure, because the antigen-specific B cells and the T cells are there, we have a very quick response, and therefore, due to this memory response, we have a quicker recovery. So essentially, what we find here is that the innate immune system does not have memory, whereas the adaptive immune system has memory. That's because these cells in the innate immune system, do not have any antigen-specific receptors or the receptor rearrangement system. So whereas the adaptive immune system, the T cells and the B cells have the antigen-specific receptors, and therefore once they leave as memory cells, they are ready to participate to the second, to combat the antigens or the pathogens during the second exposure. So basically, that is the difference between this innate and adaptive immune system. So what are the features of innate immune system? So the developmental and organizational features that the innate immune system has co-evolved with adaptive immune system. If you remember the first slide, so after that Silurian period, when we had the adaptive immune system, the labor has been shared so that the innate immune system কিছুটা কাজ করে দিচ্ছে এবং সেই কাজের থেকে কিছুটা অংশ আবার অ্যাডাপটিভ ইমিউন সিস্টেমে যে সেলস গুলো রয়েছে সেইটাকে ক্যারি ফরওয়ার্ড করছে। So basically, innate and adaptive immune system after the Silurian period have co-evolved together. And this innate immune system does not depend on immune recognition by lymphocytes, because that is the completely different cellular system, and it is functionally integrated with the adaptive immune system. Now, the cellular components of innate immune systems are neutrophils, eosinophils, basophils, mast cells, monocytes, macrophage system, dendritic cells, megakaryocyte and platelet systems, melanocytes, keratinocytes, epidermal Langerhans cells in skin, and then innate lymphoid cells in the gut. And then antigen and pathogen recognition. Innate immune responses display restricted antigen specificity. This antigen specificity is conferred by pattern recognition receptors, and germline encoded receptors are not rearranged during responses. So that is the fundamental difference that we have seen from the first slide, that these pattern recognition receptors, or the innate receptors, appeared first, and they are encoded in the germline, but they cannot be rearranged during the immune responses. So we can find out that there is B cell affinity maturation, antibody affinity maturation, which does not happen in case of toll-like receptors. And functional features, innate responses are fast, and innate immune cells participate and then innate immune responses lack memory for antigen specificity. Limited innate immune memory represents epigenetic modifications. So this is a very recent concept, that even the innate immune system has some sort of memory, and instead of the receptors, they think that it is because of the epigenetic modification of the receptors involved or encoded as in germline. So the innate immunity work as barriers. That is one modus operandi. Here we know that the innate immune system is against foreign bodies, injuries, and pathogens. Then bacteria killing substances, the antimicrobial peptides, and then protection on the outside, that is the skin, and then protection inside all mucous membranes, and first attack in the tissue is by the scavenger cells. So basically, this side of the slide you can see is the innate immune components, which come to the existence quite naturally with the growth and development, and the barriers are presented in different forms. For example, in tears, the lysozyme can, in fact, hydrolyze many different molecules. Then there is physical barrier, like in skin, the cornified stratified squamous epithelium. Then chemical barrier, like salts, fatty acids, lactic acid, and antimicrobial peptides. And then in the gut, we have rapid pH change. For example, in stomach, it is 1.50, where most of the pathogens die. And then as soon as it passes this sphincter and gets into the small intestine, the pH rises to 8.0. So the pathogens which would be adapted in acidic environment, now they are killed in this basic environment. And in addition, you have the bile salts, which also work as an antibacterial. And then in the gut, we have the intestinal lymphoid cells or innate lymphoid cells, type 1, type 2, type 3, which can combat these pathogens. And then in the trachea, we have this ciliated epithelium. So this cilia can remove the pathogens. And then we have tonsils, as was introduced yesterday by Dr. Dando. So these tonsils have these different lymphoid, what should I say, the assembly of cells, and that can take care of these pathogens. And then we have the mucous lining in trachea, and mucus and phagocytes on the innate lymphoid cells in lung, including these alveolar macrophages. And stomach, here you see I have already told that is 1.5, and then the normal flora compete with the invading microbiota. And of course, there is continuous, flushing of urinary tract so that they cannot colonize in the urinary tract. So this is basically the normal physiological mechanism that is very innately integrated into the organism, and that is what altogether presents the barriers to any pathogen invasion. Now we'll start with the antimicrobial peptides as direct effectors. So we have three different defensins: defensin alpha, defensin beta, and defensin theta. And of these, the defensin alpha produced by neutrophils. Cryptidin is another form of defensin alpha, which is secreted by Paneth cells in the crypts of Leberkuhn and galt, gut-associated lymphoid tissue. And then defensin beta is in mucosal-associated lymphoid tissue and secreted also by keratinocytes and also in lung, alveolar epithelial cells. And all of these, including cathelicidin, secreted by epithelial cells, macrophage, keratinocytes, they all integrate into the membrane of the pathogen and form pores. As a result, the cells become leaky and they die. In addition, there is histatin, which is produced by salivary glands, and they also attack the fungal membrane. And phospholipase A2 or PLA2 from neutrophils work on gram-positive membrane, and BPI neutrophils produced and work on gram-negative membrane. Lactoferrin produced by neutrophils or epithelial cells lies the bacterial membrane. C-reactive protein IL-1 and IL-6 induced from hepatocytes, bacteria, they work on bacterial membrane. And pentraxin 3 or PTX3, that is TLR induced in dendritic cells, macrophages, and polymorphonuclear cells like neutrophils, and they also work on bacterial and fungal membrane. So basically, that there are different antimicrobial peptides that can work on different pathogens like bacteria and fungus and can combat them. So the mucosal immunity at different sites in the MALT system. So mucosal surfaces are the major port of entry into the body for pathogens. So mucosal immune system protects mucosal sites in the alimentary tract, that is gut-associated lymphoid tissue, the respiratory tract, bronchoalveolar lymphoid tissue or BALT. But we need to remember that in a human, it is not active inherently as in other animals. So we have IBALT, so that when, for example, we get Mycobacterium tuberculosis invasion, that Mycobacterium tuberculosis induces BALT, so we call it IBALT, and then the genitourinary tract. So the major immune effectors at mucosal surfaces are protease-resistant secretory IgA actively transported via the mucosal epithelia, and that blocks pathogen entry. Then resident macrophages engulfing pathogens. This is, that the macrophages are in different tissues in different names. For example, the monocytes, the circulating monocytes, when they reach the spleen, that is splenic macrophages, but when they go to liver and is adopted there and become a part of it, we know that is Kupffer cells, or when it is in lung, it is called alveolar macrophages, or when it is in in brain, then we call it glial cells or microglial cells. So the macrophages have many different names depending on in which tissues they are, and in skin we call them as tissue histiocytes. And then mucus-secreting cells to trap pathogens. For example, the goblet cells in the trachea, or the goblet cells in intestine. Then innate lymphoid cells secreting cytokines, intraepithelial lymphocytes, IELs, the likes of M cells that we see in Kersh patch trapping antigens, then dendritic cells presenting antigens to T cells, and mucosa-associated innate lymphoid cells or intraepithelial lymphocytes that is circulating within the MALS system. So these are basically the armors that protect the mucosal surfaces. Here are some of the examples. For example, this is in ileum, where we have these pairs patches and we have the lymphoid nodules. You can see the germinal centers. This germinal center was very nicely described by Professor Dando and later on by Dr. Suyata Ganguly yesterday. And M cells are here. You can see that has different, I would say, interdigitating kind of membranes, which is these places or these crypts or the pseudopods. They are used for collecting these microbiota or the antigens released from these pathogens, and then transport those antigens and release towards these dendritic cells or the monocyte macrophages, which then process these antigens and present it to the T cells, and the T cells can present the antigens to the B cells, which can form then plasma cells secreting different antibodies, primarily IgA, because the T cells present here, like the intraepithelial lymphocytes or the innate lymphoid cells, they produce primarily TGF-beta. And TGF-beta is the switch factor, like alluded to yesterday. This switch factor means IgM is class switched to IgA, and the factor that is required for that switching, immunoglobulin class switching, is TGF-beta. And then here is another scheme where the APC antigen presenting cells through the T cell receptors, MHC peptide complex, activate the T cells for signal, and the second signal from the antigen presenting cells to the T cells is given by B7 CD28 interaction. And these T cells also have this CD40 ligand, the activated T cells, which can bind to CD40 on the B cells, and this provides a very strong signal to the B cells to survive, that is a survival signal. And in addition, CD40 helps the IgM switch to IgG1 and also help in producing different antibodies from these activated T cells. So basically, this is what happens in the mucosal system. For example, in the intestine, one example. And then the other mechanism for this function of the innate immune response is induction of inflammation. And inflammation is characterized by 4 features: calor, dolor, rubor, and tumor. Calor is because of increase in metabolism, so that there is transfer of energy, but it cannot go through the electron transport chain, that is uncoupling, and therefore this energy is released as heat. So that is the reason we call it calor. So the inflamed site, we always feel that it is heated. It's because of this change in metabolism. And then dollar, which means pain. And that is because of release of different chemicals, for example, histamine, substance P, etc., that excite the free nerve endings for evoking the sensation of pain. And rubor, which is redness, due to increased blood supply to the affected area, the inflamed area tissue becomes red. And then we will see that the inflamed part, inflamed tissue, is always swollen. And that is why it is called tumor. And this swelling is caused by increased capillary permeability, allowing large serum molecules to enter the tissue. And that causes increase in colloidal osmotic pressure, drawing in more fluid into the tissue, and then drawing more fluids and chemokines directed infiltration of leukocytes to the inflamed tissue. So basically, these are the four characteristics of inflammation, and these are the reasons or the mechanisms how that happens. And then this leukocyte infiltration, how does that happen in case of inflammation? It happens by migration, and that is regulated by endothelia expressed signaling molecules like selectin and adhesion molecules, because the signaling and the adhesion molecules that control migration are selectively expressed in venules. The hemodynamic shear flow is low in the venules, allowing these two sets of molecules to interact. The endothelial surface charge is also lower in venules, and therefore the negative repulsion is not there. So basically what happens as a result, the rolling leukocytes can actually afford to tether, and that is why we see this leukocyte infiltration in three different steps. The first step is rolling and tethering. So the slow rolling on endothelia and venules, and then it tethers by selectin carbohydrate interaction, then chemokine directed activation and adhesion to endothelia, and then transendothelial migration following multiple steps, chemokine directed polarization of the leukocytes. So the leukocytes, if we consider that they are round, suddenly they shape the change, and on one side towards the direction of migration, it becomes tapered. And that is what we call polarization of the leukocytes, and these chemokines actually causes the intracellular cytoskeletal elements reorganization, so that these membranes can move and get tapered at one end towards the direction of the migration. Then the pseudopods to basement membranes and its dissolution, because these pseudopods start secreting different enzymes, different hydrolases and matrix metalloproteinases that dissolve the basement membrane, and that is how then they enter the tissue. Once they enter the tissue, again they move along the gradient of chemokines and arrive at the site of infection. So that is how, when a pathogen infects a tissue, the inflammation starts. And inflammation, one part is, as we have seen, is leukocyte migration, and this is how the leukocytes migrate through the endothelial cells from the vessels, blood vessels, to the site of infection. Three major plasma enzymes control this inflammation, and these are known as fibrinolytic or the plasmin system. This is the tissue-derived plasminogen activator, converts plasminogen into plasmin that dissolves fibrin and activates matrix metalloproteinases and promotes angiogenesis. And then the kinin system is the second one. Kinin system mediators, which are bradykinin and lysyl bradykinin, which is a smaller molecule they cause nitric oxide mediated venular dilation, increased vascular permeability, and smooth muscle contraction. And then the third one is the complement system, which is the C3A, C4A, and C5A, which cause inflammatory responses by promoting chemotaxis, phagocytosis, and cellular activation. It is very important that to remember that complement system is a unique system where we will see later slides that we have multiple proteins arranged in a cascade, so that the first one, once activated like C1q, it then activates C2, C3, and then C4, C5, etc. till C9. And at each step of these activations, the response is amplified. So basically, this is another cascade system besides the blood coagulation system, where we have these cascades of succeeding activator and the responder and or the substrate, and finally we get the fibrin clot in case of blood coagulation system, and in case of complement system we get the assembly of a macrophage attack complex or MAC complex. So the third one that we have in biological system is the cell signaling cascade system, which Dr. Abra Chaudhary will discuss in the context of cytokine signaling. So basically, while inflammation is a response to tissue damage, and ultimately it ends in healing tissues. So while infection causes inflammation and damages tissues, the same is also responsible for healing of tissues and progress through. First, the bleeding has to be stopped, that is, and then there is very quick inflammatory response. The inflammatory response, how it happens, we have discussed in the previous slides. And once, as a result of this inflammation, the leukocytes reach the site of infection, they prevent the spread of pathogen and kill the pathogen. And as a result, there is a damage of tissue, damage of pathogens, there is release of debris and dead cells, dead pathogens, they are phagocytosed so that the area is cleaned, and then fibroblasts and associated cells start proliferating. This is very important that when all these things happen during this inflammation, the first three steps, acute inflammation, pathogen killing, and phagocytosis, these are all part of inflammatory cytokines function, for example, IL-1, IL-6, TNF-alpha, however, interferon gamma. But when this happens, if they continue to for a longer period, which is chronic inflammation, then we get something called delayed type hypersensitivity reaction, which is not good for the host, which means that following the homeostatic mechanisms in the body, physiological mechanisms, all these inflammatory responses has to be subsided, has to be suppressed. And that is done by the activation of the opposite system, which is the anti-inflammatory cytokines, and represented primarily by three, or actually two, TGF-beta and interleukin-10. Now, this TGF-beta is a cytokine which induces fibroblast proliferation and other cells which are required in association with the fibroblast to fill out this space, which is first filled by debris, but then turned to void by phagocytosis. So this is then filled up by fibroblasts, then the blood clots are dissolved, and this extracellular matrix is remodeled, and the regeneration of cells of the tissue by the tissue-specific stem cells differentiation, and then restoration of normal structure and functions of the tissue happens. So basically, it's a multiple steps, and we have to understand that all this happens in sequence, which means it follows a set of genetic programs. So the next that we will come to is a complement system, because the complements, like I said, are organized in three different sectors. The first one is the trigger. So the trigger can happen in two different ways. One is the classical way, which is antibody. So the antibodies, through their constant region of the heavy chain, can fix it, the complements, and can trigger the whole cascade. Alternatively, there is factor B or mannose binding lectins, which can also activate these complements, and they lead to the activation of C3 convertins, which work on the C3 component of complement and releases C3A and C3B. So the C3A is the anaphylotoxin, and so is C5A, and C3B and C5B come together, and then along with C4, they start this C5 to C9 assembly, which is the membrane attack complex, because they form pores on the membrane and cause the death or the cytotoxic effect on the target cells. So basically, what we find is that the complement system have trigger region, then the central and effector region. And what we find is that the pathogens evolved different ways to target these three different steps. But anyway, so before we go to that, the complement system's function is not only the fixing the membrane attack complex on the pathogen infected cells or on the pathogen itself, but the released components can have many other functions. For example, modulation of cell-mediated immunity, promotion of humoral immunity, then immune complex transport, inflammation, angiogenesis, modulation of tumor growth, and lysis of cells infected with pathogens. And this C3 can actually coat a pathogen or a molecule, and that can be taken up by C3 receptor, and that is called opsonized phagocytosis. So all those are explained in this slide. So basically, yes, this is how it causes inflammation by C3A, C4A, and C5A by activation of mast cell, basophil, neutrophil, monocytes, and macrophages. And then there can be phagocytosis, because like I said, that this C3b can bind to this receptor, and then there is phagocytosis enhanced, and they can cause cell lysis by this membrane attack complex, and so on. So what I have tried to show you here is the classical pathway on this side, how this C1 complex is formed, that is C1q is activated, and these are different molecules like NS1, NS3, non-structural protein 1, non-structural protein 3, non-structural protein 4A, and then other proteins from different viruses. They can inhibit this activation of C1, which is the trigger. And then there are other molecules from viruses, many different viruses, and then they target different areas or different molecules of complement cascade. For example, here C2A, C4B are targeted by these different viral proteins, and here C3B, and so on. So basically, if you go to the next step of the terminal pathway or the membrane attack complex that you can see here, here you will see again these different molecules from viruses can actually prevent this activation. And I will just skip this. Again, this SARS-CoV-2, it is shown how SARS-CoV-2 can activate complements and can lead to the lysis of the SARS-CoV-2 infected cells. So basically, that is the way that complement-mediated inflammation can be started by SARS-CoV-2 virus. And these are some of the inhibitors that can target different steps of the activation or the steps of the infection. And can be thought of as a therapeutic option. So next I'll move to the cutaneous immune response, where on the skin, different pathogens are there, virus, bacteria, fungus, and mice. And then they're taken up by epidermal Langerhans cells, and that is presented to taken to the draining lymph node, where these are presented to different T cells, and then they are activated, and then they are recruited to the site of infection. And this is at the site of infection where these lymphocytes or the T cells recognize the infected cells and kill the infected cells. So this is again, you will see this collaboration between the innate and adaptive immune cells. And this is neutrophils, which are released from bone marrow, and then they stay in the circulation for 6 hours, and within these 6 hours, they can migrate to the inflamed tissue. And after they kill these pathogens, they can again go back to the cytokine and go back to the bone marrow, where the bone marrow macrophages take up these apoptotic neutrophils to keep the balance. Similarly, the eosinophils that we know can attack the bigger pathogens like helminth, as they secrete fixed complement and secrete different mediators like lysophospholipase, peroxidase, hydrolases, and that not only kills the pathogen but also cause tissue damage. And I'll skip this, and we'll see that these basophils or the tissue mast cells, they perform many different factors by secreting histamine, serotonin that cause vasodilation or vascular leak, and at the same time causes airway constriction and increased peristalsis. And on the other side, they can also secrete platelet activating factors, prostaglandins, leukotriene C4, cytokines, and enzymes, IL-4, IL-13 that cause T cell differentiation and helps B cell response. So, and then the NK cells that has already been discussed yesterday, by secreting interferon gamma, perforin, granzyme B, they can kill the infected target cells. And all these things, all these functions are mediated by the toll-like receptors and other innate immune receptors, which show very restricted specificity for particular antigens, type of antigens. And this is here that there are toll-like receptors, there are nod-like receptors, and there are C-type lectin receptors or CLRs. And all these receptors, when they recognize their antigens, they're activated and they trigger 4 different functions: phagocytosis, autophagy, apoptosis, and inflammations. So I'll just quickly go through this phagocytosis first. The target particle is phagocytosed and fused with the lysosome. And then the phagolysosome has a very low pH where this is hydrolyzed. And then these proteins are released, or this is presented through MHC class 2 pathway to the T cells. And all these different pathogens like Shigella, Listeria, Rickettsia, Burkholderia, then Francisella tularensis, then Trypanosoma, and similarly Leishmania, the Candida, the fungus, Cryptococcus pneumoniae. So there are many such pathogens which can prevent this phagocytosis and its function to destroy them in many different mechanisms, by many different mechanisms. And then, like very much like phagocytosis, there is called autophagy, where the cells which are damaged by this infection can activate different proteins like Vacillin-1, ATG5 to ATG12, then LC3, and many other proteins, which I will show in the next slide, can actually lead to the autophagosome formation that fuses with lysosome and ultimately destroy these cells. So this is the lysosomal degradation. So this has a strong similarity with the phagocytosis, the way it is done, but this is where in autophagy, the infected cells can kill itself, can destroy itself, so that with this, the pathogen is also destroyed. So these are many different proteins you can see here, which execute this autophagy. And the other mechanism of this innate immune system is the induction of reactive oxygen species, which are nitric oxides or superoxides and others, which damages the DNA by base modification or double strand breaks. Lipids are peroxidized or lost, and then proteins are degraded, fragmented, or had many different chemical modifications, which leads to their inactivation and final degradation. So this is another mechanism by free generation of free radicals that the innate immune system works. And then they also induce apoptosis, whereby the cells are damaged and stressed or triggered by body signals to begin apoptosis, and that through different systems of enzymes finally leads to the cell breakage into smaller pieces containing cell components and destroy nucleus. And then these debris are recognized by the macrophages, and they engulf these apoptotic cells. And this is where, by these different intrinsic pathways or the extrinsic pathway, apoptotic cells release the apoptotic body, which is cleared by these phagocytes by phagocytosis. And apoptosis we have already discussed. These are just different proteins. And then there is inflammasome, which are these node-like receptors, and the most crucial one or the most famous one are the NLRP3, node-like receptor protein 3. This NLRP3 builds a huge inflammasome complex of different proteins, which you will see here, NLRP3, and then NLRP3 with another many different proteins, and all these together lead to the cleavage of pro-IL-1 beta to form IL-1 beta, which is released, and gasdermin D is released. So these are the inflammatory mediators which are catalyzed, which formation is catalyzed by inflammasomes. And these are many different regulators of inflammasomes. So here you can see how an NLRP3 activated by many different proteins. So basically what we see here, that at the time zero, when there is an antigen or a pathogen that triggers the induction or activation of innate immune cells like neutrophils, tissue histiocytes, or infiltrating macrophages, eosinophils, basophils, or tissue mast cells, and then they secrete different cytokines, chemokines, and other mediators, and they then chemo-attract the T cells to the site of infection, and these T cells are activated by these different antigen-presenting cells like macrophages and dendritic cells, which acquire the antigen by endocytosis and phagocytosis, present that to the activated T cells, and these T helper cells help the B cells producing antibody, and these activated T cells, affected T cells, can eliminate pathogens, can destroy the target tissue, for example, the cancer cells, or can execute this graft rejection, and can destroy even the cell tissues if these cells are self-reactive. So these are the four different aspects of the adaptive immunity that we also need to see when we can have different diseases. For example, in case of bone marrow transplantation, we can see the graft versus host reaction, or you can see that the tumors survive, or you can see autoimmune diseases destroying our own tissues. So we can discuss all these things at a later point in adaptive immune response. So basically, what we see here at the end, that the human immune system co-evolves with pathogens. As ontogeny recapitulates phylogeny, the innate immune cells appear first during this embryonic development, and also they appear first at the site of inflammation. Innate immune system works in different ways: physicochemical barriers, direct acting antimicrobial peptides, innate immune cells in skin and mucosal surface, inflammation, complement cascade, and receptor-mediated activation and receptor-mediated functions. We have seen effector functions of phagocytosis, autophagy, apoptosis, and inflammasome. So thank you very much.